Okay. So this next session is about modeling Li-Fraumeni syndrome. And we heard a little bit earlier this morning about a tour around the world, and now we're going to do a bit of a tour around some of the animal kingdom. I will say for the families, this may seem a bit abstract. Why are we talking about mice or zebrafish? What does that have to do with patient care? But really, these preclinical models are the first step on the way to really understanding the mutations, the impact of the mutations, and eventually, as we'll hear this afternoon, how to bring it back to the patient. So again, if you're wondering why you see some zebrafish swimming across your screen, just try to remember this actually does relate back to patient care, and it actually is really very important, and I'm looking forward to it. So again, same ground rules. There'll be 20-minute talks with 5 minutes for questions. If the speakers go over the 25-minute time period, I'll be on the side waving my hands and trying to get them off the stage. All right, so our first speaker is Professor Chang Chan from New Jersey Research Institute in New Jersey, and he'll be talking about the contribution of genetics and stochasticity to variable phenotypes in Li-Fraumeni syndrome. So the floor is yours.
Well, thank you. I would like to thank the organizers for this wonderful meeting and for giving me a chance to talk about some very new work that I've just been working on. So this is unpublished results, and this slide, the first slide here, is the motivation for the problem that I want to talk about today. It's what you see. It's what looks like what you would see as a typical Li-Fraumeni pedigree. There are individuals, for example, in the first generation, the grandmother had breast cancer age 38. She has 3 children who inherited the p53 mutation. One daughter had breast cancer 26, a son who is at age 30 who is cancer-free. Another daughter at age 26 had osteosarcoma. And in the third generation, you have children who had, one had rhabdomyosarcoma at age 8 months, and another daughter who had adrenocortical carcinoma at 6 months. And then there are other siblings, age from 4 to 8, who are cancer-free. Now, the question is, in this family, every one of those individuals, they have the exact same p53 mutation. And they are related in the sense that they share, through siblings or parent-child relationships, they share 50% of the genome. Why do you see such variable phenotype within a given family? Now, next. Okay, so this is the background to talk about what are the potential variables that may contribute to these variable phenotype. And on the left, what you see is what we know is a p53 mutation by itself is not sufficient for tumor formation. Okay? So if you look on the left, right, that's sort of like a life history of an individual. You start out as a zygote with a mutation and p53 in 1 copy. What's required is that throughout life, there are these additional somatic random events that are occurring, and some of them confer some selective advantage to a cell. And over time, when a cell accumulates enough of these changes, it becomes a tumor. Now, what are the variables that play a role in how the phenotype may occur is, well, we know genetics and environment are both important, right? So, for example, the p53 mutation itself, what the genetic modifiers that particular individual has, what's the genetic background? Because genes that are involved in DNA repair in response to stress may have different efficiencies between individuals. Well, we also know environment plays a role, but at the end of the day, right, these two things, right, they interplay. And what they lead to are these stochastic somatic mutations or epigenetic alterations. These are denoted by these lightning signs, right? So these events, right, is an example of epigenetic alteration, right, would be a DNA methylation. And the reason is that changes in DNA methylation, that sort of information is passed on through cell division, right? So it's like a somatic mutation, like a genetic mutation. So these events could include, for example, right, a loss of heterozygosity where you lose a wild-type copy of p53. It can be genes, mutations, or epigenetic mutations in genes that are involved in growth, in DNA repair, in cellular differentiation, and in chromatin modifiers. So the best way to study, for example, let me go back, right? So if I want to, my talk's going to focus on this stochastic part that plays a role in the variable penetrance. If you want to study that, what's the optimal thing you want to look at? I want to control for genetics, and I want to control for environment. So what I want to look at is, well, here's an example where I can do both. Oh, sorry, I keep hitting. Back. Okay. This is a case, possibly, right, the only case in the world of a Li-Fraumeni syndrome identical twin. Right? We did a whole genome sequence. They are genetically identical, and yet they exhibit discordant phenotype. So this is a family that I think Hani Arifin had shown very quickly in her slides. But what you see, right, are two identical twin females. They're now at age 21. One twin has had no cancer, while her identical twin had a rhabdomyosarcoma at the age of 4, and then an osteosarcoma at the age of 11, and another osteosarcoma at the age of 21. Okay? Now, you can say that perhaps her treatment for her first cancer, rhabdomyosarcoma, might have increased her risk for the subsequent, right, tumors. Fine. However, we still, are faced with the fact that they're discordant for the first tumor, rhabdomyosarcoma. And by the age of 21, we are pretty sure that her twin, who's never had cancer, is not going to get rhabdomyosarcoma, right? So how do you explain this variable phenotype even with a pair of identical genomes? Now, this is the only pair of Li-Fraumeni twins we have. But what we can do is that with the mouse model, we can create Li-Fraumeni identical twins, and lots of it, to see whether this is a unique situation or is that a common observation you would have if you were to have 100 identical twins, for example. So we went and created actually Li-Fraumeni mice. And what we did is we are creating a Li-Fraumeni mice by we're using a mutant p53, which is in the mouse is the 172. It corresponds to the 175 hotspot mutation in humans. Okay, so that's a missense mutation of DNA binding domain. And we're going to create a heterozygous mutant in a mouse, and we're going to create a hybrid background. Okay, and I'll explain to you how we do that. One of the genetic background will always be a C57 block 6 background. And we cross into 7 different other mouse inbred lines that we order from Jackson Lab. So on the right, you see the AJ, BALC, DBA, C3H. These are just names of different inbred lines, right? So by crossing that into C57, what we get is what's called the F1 hybrid. Half the genome is a C57 block 6, and the other half is one of those 7 inbred lines. And these are the numbers of mice that we get now in the F1 hybrids. So these are the Li-Fraumeni mice. We've got 30 mice in the C57 cross AJ. So again, 30 mice that are genetically identical there. And those are the numbers. The ones that I'll focus on are the last two, the SWR and the NOD. So remember, that's SWR cross C57, NOD cross C57. The reason is the numbers, we have the largest numbers there, 71 and 76. So in each of those, right, remember, these are now identical mice. So we're studying 71 identical mice and 76 identical mice in 2 different backgrounds. And I'll show you the variable phenotypes that we observe. So first, right, mice is not the same as humans. And what's the main difference is that in mice, they don't get mammary tumors, right? And across all of these different, 7 different backgrounds now, in these mice, there's no, right? The other thing, right, is that we don't look for brain tumors or leukemia in the mice. So we don't know whether they exist, but it's something we don't see. The tumors that's most common is lymphomas and osteosarcomas. Okay? So in humans, lymphomas are not so common, but osteosarcomas are. And also all the other types of sarcomas are common in mice. So they do share a lot of phenotype with humans. Now, sure enough, genetics does play a role. And the role that genetics plays is that in the SWR hybrid background, there's actually, this is very unique, there's actually an increased female embryonic lethality. In that, which we don't see in any other backgrounds here. Okay, that's what interesting. And in the females that do, that are produced, they also have a higher tumor penetrance. The other difference is that in the AJ hybrid background, there's an increased incidence of lymphomas and a decreased incidence of lymphomas and an increased incidence of liposarcomas. And in the BALC background, there's an increased incidence of angiosarcomas, right? But now let me show you the real shocker. Where, so yes, genetics, changes the incidence rates usually by a factor of 2 or so, but what's shocking is the heterogeneous phenotype still. So on the top row, what you see is the distribution of age at which these mice die. So let me step back and explain to you what we did with these mice, right? So once we created these mice, we just watch and wait for them to get a tumor or to die. And when that occurs, we do autopsy and find all the tumors that they have. So what you're seeing now, right, is the age of distribution at which these mice die. And now remember, these mice, right, is genetically identical. On the left, we separate the females from the males just to see whether there are differences. And there are no differences. So what's remarkable is that there are mice whose genetic background is identical. Some are getting a lymphoma at 5 months. Another mice is not getting anything until a lymphoma at 20 months. Some mice, right, will instead get an osteosarcoma at 10 months. And then another mice might get an osteosarcoma at 19 months. And yet another mice might get an adenocarcinoma at like 15 months, right? So it is extremely heterogeneous, which is telling you is that these stochastic events that are playing a role in the life history of each one of those mice plays a very large role in the variable phenotype. And on the bottom, it's just to show you the same thing occurs with the not F1 background. This observation we observe across all the 7 different genetic backgrounds that we're looking at. So now what I want to show you, right, is how I'm going to construct a stochastic model for tumorigenesis, right? So the key component that we need to look at is a probability, right? Because probability is really the quantification of uncertainty, of randomness. So what I would like to know, for example, is it possible to determine, right? I want to focus on humans, right? For a Li-Fraumeni person, right? What is the probability of getting a specific tumor type as a function of age? That's really the central piece of information that I want to compute, if possible. So what I'm going to do now is, and the great thing is that Pierre has already introduced this database, the IOC database, which is, and the thing that I did with this database is that I removed the R337H mutation. That's the single mutation that was the highest frequency. It was a Brazilian cohort, due to a founder mutation. It may behave differently, so I removed that. And I'm looking at the rest, and so these are carriers of mutations that are primarily now, found with a distribution that is mostly in the DNA binding domain, in the central core. Now, with every database, there's obviously all kinds of complexities of ascertainment bias, right? For example, in this database I'm working with now, there is 1,000 women compared to about 700 men, right? And that's probably due to some ascertainment bias related to the fact that women may be getting tumor at a higher rate, so that they get selected for in this database, right? So here is now the first thing that I would say, right, is the biggest difference that you see when you look at the incidence rates, right, is really sex dependent, right? Once you remove, let's say, the sex specific tissues, even for adrenocortical carcinoma, right, females are at 2 times higher. Well, this is incidence, right? The percentage of individuals having these types of tumors. For adrenocortical carcinoma, females are more likely to get it, whereas for all the other tumors, males are actually more likely to get that. So, for example, osteosarcoma, leukemia, lung, stomach, and brain cancer. And also the early types of soft tissue sarcoma, males are actually a higher rate of getting that. But again, right, this is incidence, it's not probability. I want to construct the probability of getting tumor as a function of age. So So before I get there, right, let me tell you that in this database itself, this is the overall survival curve for Li-Fraumeni syndrome, males and females and males. So as you can see, right, with this selection of individuals in this database, by the time you get to age 65, there's 60, there's about less than 10% of the of both men and women who are still surviving at that age. So here's what I'm going to do. I'm going to on the first distribution that you see, right, on the top left, right, that is the incidence rate of breast cancer as the first tumor type in a woman. And I'm only going to focus on the first tumor type because treatment may impact their likelihood of getting subsequent tumors, right? So if you look at the first tumor that they get, we know that treatment is not affecting that. So that's the incidence, and it's very, it's well known that a lot of it is occurring, between the age of 20 and 40, so premenopausal. But the fact that it gets very low as you go to longer to old age, right? It's really right due to the fact that there are very few women who are alive at that age. So that's why the incidence is low. So to correct, right, if you want to have a probability for getting breast cancer at age 65, you have to correct for the fact that there are a very small number of women there. So I use the overall survival curve to correct for that probability. And what you get on the lower left plot now, right, is actually the probability of getting breast cancer as a function of age, right? So you see that, it doesn't go down the way you would, at old age. However, the only caveat now, right, is that beyond 65, the number of samples you have is small, and so the errors can become large. So for my modeling going forward, I will study everything up to the age of 65. So up to the age of 65, that's the plot you get on the lower right panel. That's the probability of getting breast cancer as a function of age up to age 65. So I now do this, right, for every cancer type: soft tissue sarcoma, brain cancer, adrenocholangiocarcinoma, ascertainment. So these are now, right, not the incidence, but the probability of getting those tumor types as a function of age, right? And so now, right, when you do this for the common types of tumors, right, at least up to age 65, right, you see that most of these probabilities, it does increase with age, like everyone else in the general population. And so that was for women, and these are the corresponding probability curves for men, right? So now, what I do is that I use these probability curves to actually compute what the lifetime probability of getting tumor is, right, up to age 65. So now what we find is that for women, to get any cancer up to age 65, you have a 90% probability of getting any cancer. For breast cancer, lifetime probability is 76. Soft tissues, 23%. Brain tumors, 17%, right? And so on. Adrenocholangiocarcinoma is only 7%. 7% lifetime probability of getting the same for the osteosarcoma. For men, from this database, the lifetime risk of getting any types of cancer to age 65 is 88%. Brain cancer is 30%. Soft tissue sarcoma is 26%, et cetera, right? So this is now going to be the key to explaining why we see heterogeneous penetrance even for individual families. And the reason is, right, when you talk about penetrance, you need to define what phenotype you're looking at. If you want to define a phenotype as getting any cancer, then there's high penetrance, right? But if you want to define your phenotype as getting, let's say, a very specific tumor type, for example, adrenocholangiocarcinoma, the penetrance for Li-Fraumeni is only 7% if you're female, and if you're male for adrenocholangiocarcinoma, it's only 4%. That in itself really explains why we see the heterogeneous phenotype within a family. Even with identical p53 mutation and with identical genetic background, if you're only having a 4% chance of getting a tumor, it's unlikely that your identical twin will get the same tumor type. So here, right, the key, so what is the probability distribution teaching us? One thing it's teaching us that the shape of the probability distribution, right, really tells you about the biology of the tumor type, right? So I gave you as like an example, right, 3 different tumor types with very different distributions, right? Adrenocorticocarcinoma, right? The middle panel is osteosarcoma, and the right is a common tumor. I think it's, I can't see what it is from here, but it's the common types of tumor that increases with age. So what tells us is that for each of these tumor types, these probability distribution tells you that the pool size of susceptible cells, right, it changes over time. There are some types of tumors, right, that are probably coming from embryonic types of tissues that over a certain age, you don't have those tissues, they go away so that beyond a certain age, you'll never get the type of tumor, for example, right? And also, the probability, right, it depends on the rate and the number of mutations of these additional mutations that are required for tumors to occur. So there's a lot of biology that's involved here. But again, right, the thing that I want to emphasize here, right, is that what is striking is the contribution of the stochastic events over the genetic modifiers that we've always been thinking about as geneticists, right? It's really astounding. So that here to summarize, right, the explanation for variable phenotypes is that because every tumor type itself has very low probability, right? That is what is the basis for why you get different tumors occurring even within the same family. So now, right, what can we do with these models? And here, I'm just going to go very quickly, right? I mean, because it's not polished enough to really tell you, right? So, for example, in the data, right, we can look to see, right, are there tumor type dependencies, right? So, for example, right, in this, right, you have women who get both breast cancer and a soft tissue sarcoma. There's about 4%. The percentage that get, let's say, breast cancer and a brain cancer is much lower, 0.0097%. I mean, sorry, that's not percent. That's actually so 0.043. It's 4%, right? So the question is, right? We have this data, and the question is, we're looking for patterns to see whether certain pairs of tumors are occurring more often than not. Since we have humans tend to have very bad intuitive feel for probabilities, modeling is crucial to really help you look for patterns. So we can use the model, stochastic model, right, to really generate 1,000 individuals based upon the probability distributions of getting various tumors. 4 minutes, and so we can essentially, right, going fast forward, right, we created a Monte Carlo simulation. We can generate, for example, 1,000 individuals based upon the probability distributions that you see on the left. And another crucial element is that in the data you see, most individuals who are diagnosed with the first cancer, right, their life expectancy of that is very short. They actually die, in this data set, most die within 2 years, actually. So we're to incorporate that element into the modeling, that when we simulate that when they get the first cancer, we also have this simulation as to how long they live afterwards. So now we can create, this is, on the bottom is the simulated data, and based upon simulated data, we actually see there are certain pairs that occur at higher incidence and certain pairs that don't. And the higher incidence, right, it shows that there are certain tumor dependencies which suggest that there may be genetic modifiers that affect both types of tumor tissues, or perhaps, in this data set, perhaps there's a treatment that predisposes them to a certain types of, subsequent tumor as well. So that's the usefulness. Now, to go back to the identical twin that we have, right, we can now ask a different question. What's the probability of a Li-Fraumeni individual having 3 cancers by the age of 20? So we simulated 1,000 females, right, based upon the probabilities that I computed, and it turns out, right, that if you simulate 1,000 females, by age 20, 70% of them, right, would not have any cancer yet. 25% would have had 1 cancer. 4%, right, would have had 2 cancers. And you see, right, 0.003, right, less than 1% would have had 3 cancers. So the likelihood of that individual having 3 cancers by age 30, right, it means that those 3 cancers, right, are not occurring independently, right? It could be that in her case, right, the treatment for the first cancer predisposed her to having the subsequent 2 cancers as well. So these are the sort of the usefulness of this kind of stochastic modeling that we can do, so that we can ask these questions. So I'm going to skip this slide, and I'm just going to summarize here again that what I want you to leave with, right, is that if you knew someone's genome, it is still very hard to predict what their life history will be. And that's because for tumors to occur, these stochastic life events are the things that are being selected for that are necessary. And there's a large amount of unknown involved in that. And what that means is that, there are certain things that are very challenging, to study from the point of genetics. I think that the things that are useful, for example, is what Pierre shows, that the p53 specific mutations may alter the probability. Some of the genetic modifiers that directly interact with p53 may alter that. But these alterations, right, for a tumor type, right, even if it affects by a factor of 2, right, are pretty moderate in that case. And I think that what's very useful is actually to go after those life history events that are occurring. So for example, if we can identify what some of those important life history events that are necessary for tumor formation, they can serve as amazing biomarkers. And knowing those biomarkers and being able to detect for them will drastically improve your probability, what the subsequent probability is for getting, an additional tumor. And really, so what I'm saying is that the key to do that, to find those early events, those potential biomarkers, we need to sequence a lot of Li-Fraumeni tumors. Right? So I want to end there, and I want to actually acknowledge. So the person who did the stochastic modeling is Huaka. Yvonne's son is the one who actually did all the mouse models. And I had to acknowledge my collaborators and my mentor, Arnie Levine. I've learned so much from him. I've also learned so much from Pierre. So thank you very much, and I'll take any questions.
Thank you very much. I like the hypothesis. I think there has to be stochastic events, and I think that explains variation in other identical twins. We in fact have got an identical set of twins with p53 mutation. One of the twins had radiation treatment of a hemangioma and developed 3 sarcomas in the radiation field, age 3, 10, and 13. His identical twin didn't develop his first sarcoma until 39. So clearly, I agree with you. I think the first one may be stochastic, but after that, it's potentially related to the treatments that they may have had.
So, I also have a lot of questions, but we're out of time, I'm afraid. But there's plenty of time during lunch to follow up with the speakers. So how about another round of applause? Okay.